So, just to recap, because it's really, really important, the evidence that existed because of the that support the development of photosynthetic prokaryotes, which began the production of oxygen in the atmosphere. The first one, so I would start making a list. I'm going to try to write as best as I can. Still didn't get my my thing. So the first one, number 1, is the presence of rusted iron in rock. So I would write presence of rusted iron in rock. Again, and these fossils with rusted iron done by presence of oxygen can be dated to when this occurred. The second form of evidence that we have is when they find or have found either evidence of chlorophyll and chloroplasts in rock around prokaryotes that they find. Again, the evidence of chlorophyll signals the ability to absorb energy from light. And again, once you be able to absorb energy from light, if you remember from photosynthesis, water is split into oxygen. So those two forms of evidence, the presence of rusted rock, which is able to be dated, and the presence of either chlorophyll or chloroplasts in fossils found around this time, is evidence that it was the prokaryotes that evolved the ability to do photosynthesis that caused the development of oxygen in the atmosphere and led to the very disruptive but very beneficial oxygen revolution. Now, the next event in the history of the Earth is the formation of eukaryotes. And it's believed what happened to form the first eukaryotes are sort of two major events. The first one is that the prokaryotic cells began to invaginate their membranes, making things like the ER, the nucleus, the Golgi apparatus. And again, they found this beneficial because if you remember, compartmentalization is a beneficial thing because it increases the efficiency because particular things that are specialized can occur in those places. So that's what's believed happened, is that the cell membrane, remember, which was developed with the protobionts, began to invaginate, creating these compartments. And we have like the ER, we made a compartment around the DNA to protect it, we made Golgi. And it's believed that because of this increase in efficiency, that these infoldations were beneficial and then stayed in the population. The second event, this is also really important, the AP exam loves it, is the idea that these new sort of prokaryotic cells that became eukaryotic cells with invaginations engulfed other prokaryotes that looked like or had the ability to do what our mitochondria can do. So it's believed that the mitochondria was once a free-living prokaryote that was able to use oxygen, and that our prokaryotic cells engulfed them, taking them into their cell and becoming part of the cell. But the idea is that the mitochondria was once a free-living aerobic bacteria, meaning they could use oxygen, and that it was engulfed by prokaryotes that had these membranes, these invaginated membranes, and that relationship was beneficial, and therefore it stayed in the population. It's also believed that this happened with chloroplasts, that chloroplasts were once free-living photosynthetic bacteria, and that a cell with a mitochondria engulfed it. And that this relationship between the now sort of eukaryotic cell and the photosynthetic bacterium that was engulfed was beneficial. Therefore, it stayed in the population. This theory, which is one of the first theories founded by a woman, is called endosymbiosis. Okay, and endosymbiosis is the idea that both the mitochondria and the chloroplasts were once free-living bacteria that were engulfed by prokaryotic cells with invaginated membranes, and they became part of them because the relationship was beneficial. Now, once again, we need to go to the evidence that supports this theory of endosymbiosis. The first form of evidence is structural evidence, and it's the idea that the mitochondria and chloroplasts look like bacteria. They have a very similar structure to early bacteria on the Earth. The second form of evidence is genetic. And that's the idea that mitochondria and chloroplasts have their own circular DNA like bacteria. They also have the ability to move freely within the cell and also reproduce independently of the cell. In all sort of forms of evidence, they act like they're their own organism. So the forms of evidence are, again, the fact that they look like or resemble bacteria. They have their own they have their own membrane or lipid bilayer. They have their own DNA. And they're able to both reproduce and move freely without the help of the cell. All those forms of evidence support the idea that mitochondria and chloroplasts were once free living organisms that were engulfed and became part of a eukaryotic cell. And again, this theory, which was founded by this woman, Lynn Margulis, is known as the theory of endosymbiosis. Now, if we look at the tree of life, we look at the three domains that I talked about on Monday. We have bacteria and archaea. And these are two forms of bacteria. Bacteria, this domain, is the bacteria that we know of. Archaea tends to have more extreme bacteria that live in extreme conditions, particularly methogenic bacteria, or an example, that live in hot springs in the Grand Canyon and things like that. These are more extreme bacteria that live in extreme environments. And then we have us, the eukaryotic cells. So it's believed here the common ancestor of all life is the early prokaryotic cells that evolved from the protobionts. Then it's believed that there was a split that occurred, okay, where bacteria stayed, with the prokaryotic organism stayed. But there's a split that occurred in the history of life that caused the development of eukaryotic cells. And the main event that's thought to have caused that is when prokaryotic cells engulfed mitochondria. That causes the split between the bacteria and the archaea and eukaryotic cells. Now, the second event that occurred, which is number 4 here, is when the now eukaryotic cell that had mitochondria engulfed a chloroplast. And that led to our current day plants here that have chloroplasts and mitochondria, and also where those that did not get the chloroplast, our animals here, and protists and fungi, but all the non-photosynthetic organisms here. But this is the tree of life, the idea that these events, the engulfment of a mitochondria like prokaryote, and the engulfing of a photosynthetic or chloroplast like bacteria, caused the split between plants-like organisms and animal-like organisms. So, let's go back. After the development of single-celled eukaryotes, we get the development of multicellular eukaryotes. And again, the thing that evolved that caused the development of multicellular eukaryotes was the ability to do cell communication. That is by far the most sort of important event that led to the development of multicellular eukaryotic organisms. We're not going to go into detail of this because, again, they usually don't test it on the exam, but there's an event that occurred after sort of the development of multicellular eukaryotes, which is the diversification of animals, which is known as the Cambrian Explosion. And during this time, which is about 543 million years ago, we get a huge development of all these sort of different groups of animals. We see the development of sponges, chordates, organisms with the backbone, earthworms, huge sort of diversification of the number of animals on the Earth at that point. Again, and what is the evidence for this sort of development of both multicellular eukaryotes and also the diversification of animals is that we find these remains in fossils. So that is the sort of main evidence about when these occurred. Again, we're not going to go into the selection pressures that cause this, but the fact that the evidence for this event is found in fossils or appears in the fossil record. And again, those fossils can be dated. All right. Well, enjoy the rest of your snow day. Remember to answer the homework, which is essentially to explain the events of the history of life on Earth, and then record or remark the category of evidence and that is used to support that event on Earth. But I will see you guys hopefully on Thursday, and maybe tomorrow, but we'll see. All right.